All right, let's go ahead and get started. Hi, everybody. Glad to be here for the last session. My name's Holden Maker, and I'm one of the ITI faculty. So it's my pleasure to chair this session, beginning with Michael Fischbach's talk. So Michael received his PhD in chemistry from Harvard in 2007, then did an independent fellowship at Mass General before joining the faculty at UCSF. Where he was before moving to Stanford in 2017. He's now the Liu Family Professor of Bioengineering at Stanford and Director of the Stanford Microbiome Therapies Initiative. He's a recipient of the NIH Director's Pioneer and New Innovator Awards, which is pretty impressive, and also a whole list of other awards and fellowships that I won't go through. His laboratory studies mechanisms of microbiome host interactions, and it's a pleasure to have him talk to us today. Thanks, Michael.
Thank you, Holden. And yeah, delighted to be here to share a crazy story with you guys. Did not intend for any of this to happen. So yeah, true surprises sometimes still do happen in science, and this is one of them. I'll tell you how it came about. I'll frame it like this. If I were to ask you, what do you think the immune system means to do? What is its purpose when it sees a pathogen? We could discuss and debate, but we would end up agreeing probably that it's there to limit replication and dissemination so you don't die. If I were to ask you, what do you think the immune system is trying to do? What's on its mind when it sees a microbial colonist that lives on the skin or in the gut? I don't think you would know the answer. I certainly don't. This is puzzling, right? Because these are our friends. So if I were to tell you that the immune system doesn't see them at all because there's a barrier in the way, or somehow it does see them, but it knows they don't mean any harm, it gives a friendly wave and walks in the other direction, then you wouldn't be surprised. But that's not at all what happens. So I'm going to try to convince you that what does happen is bizarre and demands explanation and could end up being useful. Okay, so everything I'm going to tell you that does happen, I'm going to explain in the context of the T cell response. Why? Because the labs who did the seminal work in this area, all of which happened before we got involved, were all T cell labs. No other better reason. Certain bugs, when you put them on the skin or in the gut of a mouse, lead to a big burst of T cells on the other side of the barrier. 4 strange things about those T cells. And the first is that the flavor of T cell it's elicited seems to be dictated by the bug who's doing the eliciting. The mechanism of this is completely unknown. And I'm going to spend the rest of the talk going through the odd case of this ubiquitous skin colonist named Staph epidermidis. Every square centimeter of skin on every human on the planet is colonized by Staph epidermidis, which elicits CD8 T cells. Not known why. Second thing is that the T cells that come out the other end of this process are, in every case someone has looked carefully, antigen specific, meaning they express a TCR that's hunting for a peptide on the bug who's doing the inducing. So this is not simply a case of bugs turning up or down the volume knob generally on the immune response. This is an antigen specific T cell, the kind of specificity you wouldn't be surprised to see in the context of the response to a pathogen. But remember, this is happening in the context of the response to a colonist, which leads me to my third point, which is that textbook immunology would say that to get an adaptive immune response, you need a breach in the barrier, an infection or a needle poking through your skin during a vaccination. 5-alarm fire, innate immune cells rush in, there's swelling in the tissue. Hopefully that innate immune response summons an adaptive immune response that clears the infection. This process on the right, by contrast, looks completely different. So you put a bug on one side of the barrier, and if you look in the tissue, you see nothing. There's no inflammation. There's no influx of anything. It looks like all is quiet. It's just that about 10 days later, a big burst of T cells show up on the other side of the barrier. And perhaps most puzzlingly, they don't seem to do anything. They just loiter in the tissue or in the draining lymph node near the site of colonization. Okay, so before I tell you sort of the main thing I mean to say today, I'm going to tell you that we had one clue about what was going on here from work that Erin did while she was in my lab. So I'm going to explain what Erin did, And then I'll tell you why she did it, because even though that's backwards, it works better in that order. So Aaron had originally been interested in understanding the mechanism by which staph epidermidis colonization leads to the induction of CD8 T cells. Maybe there's a molecule on the surface of staph epi that helps to elicit CD8 T cells or something like that. But in the context of her work, she became more interested not in the mechanism of how, but why. What is the purpose of this process, or why does the immune system respond this way? And so the way Aaron probed that was to do the experiment on the bottom of the slide. What she did was to engineer staph epidermidis. She had developed a genetic system for this bug, which was difficult to manipulate otherwise. So she developed having developed this genetic system, she engineered staph epidermidis to express a tumor antigen on its surface. Why did she do that? Because she wanted to trick the immune system into eliciting CD8 T cells that it thinks are specific for the bug, but we know are also going to be specific for a tumor that resides elsewhere in the mouse. Why did she want to do that? Because she wanted to put these CD8 T cells to a functional test. Two diametrically opposed possibilities. One is that they would do what everybody had observed them to do to date, which was just loiter near the site of colonization and do nothing. The other possibility, of course, is that they would do what any self-respecting CD8 T cell should do, which is to leave the site of colonization and go to the tumor and kill tumor cells. Two completely different options. So the experimental setup is very simple. This is not like other work in my lab that some of you have heard about. These are immune competent wild type black 6 mice, straight out of the shipper from Taconic. Aaron colonizes first and then injects tumor cells a week later. Why did she do it in this order? Because she didn't think this was going to work. So she wanted to give the colonization process a head start, as it were, and didn't prepare the mice in any way. When you colonize these mice, no shaving, no scrubbing, no adjuvant. All that happens is she dips a Q-tip in a bacterial culture and rubs it gently on the head of the mouse. That's the whole experiment. So in this experiment that Aaron did right from the start, this is what we saw. This is an aggressive syngeneic tumor model. So if you don't treat the mice at all, or if she colonizes them with a wild type strain of staph epidermidis, all the tumors go out of control and all the mice die. Whereas if she colonizes with her test article, which was a pair of strains, each making a different form of ovalbumin, then the tumors completely go away. There's a bunch we could talk about that sort of followed from this result, but the paper's published, and the reason I'm telling you this is so that you can understand why Jeanette did what she did. So Jeanette was working with Aaron on this project. She's an immunologist, she was trained as an immunologist, and had just gotten started as a postdoc in my lab. So started out by learning the ropes with Aaron. And this didn't add up for Jeanette. Didn't add up for any of us, to be honest. We had all been talking about the T cell response to colonists as being somehow different. We used the word homeostatic a lot. We felt like it was neutered or anergic or somehow weaker than a normal T cell response. And so seeing that the T cells that are elicited by staph epidermidis are capable of taking down a very difficult immunologically cold tumor made us think that there might be more here than we had realized. Okay, so in light of that, and I'm going to explain Yasmin's contribution in a moment, Jeanette asked a very simple question, which is that reason that when the adaptive immune system goes after something, it's not just T cells that are employed, but also B cells, which make antibodies. So she asked, might there be an antibody response against this bacterial colonist on the skin? I have to insist, maybe in my own defense, that this is a ridiculous question. There's a skin barrier in the way, and why would the host mount an antibody response against a colonist living harmlessly on the skin of every human on the planet? It doesn't make any sense. She still did the experiment. So experiment is very simple, just like Aaron's. Jeanette took a Q-tip and dipped it in a bacterial culture, rubbed it gently on the head of a mouse. I don't have to remind this crowd that sort of the kinetics of antibody responses that Jeanette and I were used to seeing in papers that we read during the pandemic is there'd be a spike of IgG in the bloodstream, and it would go back down, and then you would wait a few weeks and give a boost dose, and then it'd go up a little further and come back down. But that's not what happened here. What happened here is sort of I'd say two things. One is that IgG in the bloodstream grew in, and it just went up and up and up until we reached about 6 weeks, where we were getting titers of around 10 to the fifth, which is very strong. We would have been delighted to see titers that high if we had immunized the mice using some conventional means. That's number one. Like super, people had characterized other antibody responses against commensals, for example, in the gut. Nobody in the field has ever seen anything nearly this strong. So I think the magnitude of this took us by surprise. And then it took a little while for us to realize this, but the second thing that took us by surprise is that the antibody never goes away. We've now repeated an experiment that Yasmin did. 9-month time point, which I just saw, the antibody levels are completely flat at around 10 to the fifth, haven't gone down a bit. So it is as though encounter with this colonist leaves the mice immune to it, which is a funny thing to say. Okay, so it's around this time that we learned, and Yasmin learned, that we were both working on the same story. We both had exactly the same data. This was great because Yasmin's a very close friend and we had been collaborating for a long time. I'll put her picture back up so we can talk about her. She did a couple of things that were really helpful to us. One is that she, for that original story I just talked about briefly, took Erin into her lab for several months and showed her the ropes, everything she needed to know in order to run all this kind of work in my lab, which was really generous of her. And then second, once she and I learned that we were working on the same thing, she was extremely easy to work with. We kind of had a meeting every month and coupled the stories together, but went off in different directions, ended up co-submitting them, and they were published back to back. And so I'm going to tell you what Yasmin found, because I think I'm going to mention it now. I'll come back to it at the end. I think it's very important for understanding the mechanism by which this happens. Two things. One is that the process of colonization on the skin leads to the formation of a tertiary lymphoid structure right underneath the site of colonization that in every way resembles a germinal center, and that structure seems to be responsible for making antibody. It's not the only place antibody is made. I think it charges the local tissue with antibody, and that conventional germinal centers in the draining lymph node are also making antibody, and they're responsible for the systemic antibody response. That's number one. Number two is that she found that there's one cell type that's required for this to happen, and that's the Langerhans cell, which, as many of you know, has these finger-like dendrites that come off the cell body and are known to kind of poke between tight junctions in the epithelial cells that line the hair follicle to reach to the outside. And when Langerhans cells get activated, they leave the skin and go quickly to the draining lymph node. And I think that process has been a little mysterious, and so I'll have more to say about that at the end. Okay. Jeanette's story went off in a different direction. First, and I'll just park this here as a little island, but it's something that we've come back to since the paper is published. Jeanette wanted to know, is there any chance that any of this might be conserved in humans? So, okay, let me tell you what she wanted to do. What she wanted to do would have been to take Bali and swab his skin and isolate his staph epi, and then draw his blood, and then see whether he has antibodies in his blood against his own colonists. That would have been the experiment, the ideal experiment. But we were in a rush, and instead, Jeanette just got 10 or so blood samples from the Stanford blood bank, healthy random adults, and ran an ELISA from those blood samples against the staph epi strains that we had in the freezer. So not a perfect match, but good enough to ask an initial form of the question. And we were quite surprised to see the data I'm showing you here. You can't find a human, we can't find a human who doesn't have really high antibody levels against staph epidermidis. Again, this would be a delightful thing to see if you had just vaccinated these people with staph epi, and needless to say, none of them have been vaccinated in this way. So this doesn't guarantee in any way that the stuff I'm about to talk about also happens in humans, but I think it makes it more likely that something like this is happening. Okay, so what did Jeanette want to do? She wanted to clone the target of the antibody response. This is an innocent question. It's the kind of thing people in my lab would do just to understand mechanism on the microbial side. And we knew that antibodies were sticking to something on the surface of the bug. So she devised a clever experiment, colonized mice with wild type staph epidermidis. We knew by then that this leads to the induction of a strong antibody response that's systemic. And so she took those antibodies that are induced by the process of colonization and asked, do they still stick to the surface of a series of staph epidermidis mutants that are each missing one structure on the cell surface? She figured if she found a mutant to which the antibodies no longer stick, then that would indicate that she had found the target of the antibody response, or at least the class of thing that is targeted by antibodies. So for example, the LGT mutant has no lipoproteins in the plasma membrane. The DLTA mutant has no dealanine modification on teichoic acid, and so on. So as it happens, she did find a mutant to which the antibodies didn't stick well. That mutant is missing a gene called SRTA. That gene encodes an enzyme that many of you already know called sortase. Sortase is an enzyme that's responsible for putting proteins on the gram-positive bacterial cell surface. If you have a protein on the outside of a gram-positive bacterium, got there by way of sortase. And so that made Jeanette's life a lot easier. Two reasons. One, you can computationally predict substrates of sortase because they all have a conserved domain near their C-terminus. It's actually the recognition sequence for the enzyme. And second, there aren't that many sortase substrates in any gram-positive bacterium. There are only 9 collectively between the 2 strains of staph epidermidis that Jeanette was working with. And so that enabled her to narrow down pretty quickly to the protein that we think is not the exclusive target, but is the predominant target of the antibody response, a truly strange protein named AAP. My favorite picture of AAP is this one. It's not ours. This was in the literature. These hair-like projections coming off the cell surface are AAP. So what is this protein? It is a giant, roughly 150 kilodalton protein that's entirely composed of repeat sequences. The green and yellow domains are direct amino acid repeat sequences. They are about as tall as peptidoglycan is thick. So they stick straight up through the peptidoglycan like the trunk of a tree. And they're there to display this red domain. This is not a direct amino acid repeat, but it's a structural repeat, as you can see. I would call this a beta solenoid. It's got 3 sides, triangular like a prism. 2 sides are very highly conserved, and 1 side is hypervariable, like this domain is meant to stick to something. And then the blue domains, we can't even get a structural prediction for them. But if you get rid of them, the protein doesn't express. So I think there's a couple things to say about this protein. One is that my B cell friends have told me that if you wanted to drive a B cell crazy, the best way to do it would be to put something composed of repeat sequences way up high where it's easy to bind it, because then you can line the B cell receptor up along it and form a very strong locus for signal transduction in the B cell. So it almost seem like this bug is trying to provoke an antibody response. This protein, or members of this family, are highly conserved, not just in staph epidermidis, in fact, not just in other species of Staphylococcus, but also in just a wide variety of gram-positive colonists of hosts. So it seems like this is not the only case of this, and we're not sure why that happens. Okay, so having found that this giant tree-like protein, AAP, is the predominant target of the antibody response, this gave Jeanette an idea. She wondered, since we're eliciting a pretty strong antibody response against this red domain, what happens if we chop it out and replace it with something that we want an antibody response against? She chose tetanus toxin, or the C-terminal fragment of tetanus toxin that people use in vaccines, which is called tetanus toxin fragment C, or TTFC. And we're going to color that orange. So schematically, what she's hoping to do is put something non-native on the surface of the staph epidermidis cell, displayed in that tree-like protein AAP, and colonize a mouse and ask whether the immune system develops an antibody response against the orange thing, against tetanus toxin. That's the idea. So she tried a few constructs that didn't work, and one that did work was just a clean swap of this red domain for tetanus toxin fragment C. So in the experiment that I'm showing you here, we're comparing 2 strains, one in which staph epidermidis has been engineered to secrete full-length tetanus toxin fragment C. And the second in which the staph epidermidis has been engineered to express a variant of that tree-like protein AAP that has tetanus toxin fragment C stuck into the middle of it. So she colonizes mice with both of these strains, Q-tip on the head, like we talked about before. And now we find, and again, this was quite surprising to us because, I mean, we could go through this in more detail, but this is a difficult protein engineering problem. It's like 150 kilodalton protein that has to snake itself through a pore, single file, and refold autonomously on the outside of the cell. So we didn't think any of this was going to work. But lo and behold, we get an antibody response as a titer of about 10 to the fifth now against tetanus toxin. And based on literature precedent, we figured this is probably 100-fold in excess of what we needed in order to render these mice immune to a lethal challenge with tetanus toxin. And so Jeanette did that experiment, and we found what we had anticipated, which is that mice that have been colonized with this strain were all protected from a lethal challenge, whereas mice that were in the control group, who would have been immune against diphtheria, all died. And I'm going to show you a video of this because this is when we realized that we were working with something that was more powerful in some sense than we had thought. The mice on the left have just been injected with tetanus toxin. They're shivering, they're having trouble breathing. This is what tetanus toxin does. And within 90 minutes or so, all of these mice will have died. Whereas the mice on the right were also injected with tetanus toxin, but they were in the treatment group, and you can see they show no signs of trouble. Remember, all that happened to them was the Q-tip on the head. So it was a very strong physiologic response from a very gentle intervention. Okay, so at this point, Jeanette and I got into a bit of a disagreement. Jeanette was happy with these results, as was I, but she felt that this approach wouldn't generalize very far because in order for an immunogen to work in this context, it would have to be something that can be produced in a bacterial cell, folded properly, post-translationally modified, and that it's really limiting. You're not going to get a viral glycoprotein made properly in staph epidermidis, not without some substantial engineering. And so she wanted to figure out a way to make an antigen recombinantly somewhere else, like in a mammalian cell culture, and then click it onto the bacterial cell surface. I felt that was a terrible idea because then the bacterial cells would divide, the antigen would dilute out, and our effect would be weak at best, maybe nothing. Where Aaron had come up with this really elegant genetic system that caused the bug to sit in the site of colonization and ping the immune system over and over again, and that this was the source of the strong response we were seeing, and that why would we do something inelegant like Jeanette's idea? This is a terrible way to end a paper. We went around the horn a few times on this. Jeanette suggested it at least 3 times, and I said no at least 3 times. And so Jeanette went downstairs to my friend and neighbor, Christopher Barnes, who told her this was a wonderful idea, and that, and in addition, if she was going to do this, that what she should do is to put spy catcher, system I don't need to explain to you guys, into AAP, and then use spy tag antigens. They should just click onto the bacterial cell surface. So Jeanette did this. Her first experiment was with spy tagged GFP, and in this sort of plot, you can see that works. We do get GFP covalently attached to the bacterial cell surface. I started paying attention here because we estimated that the copy number was around 40 to 50,000 copies of GFP per cell, which is a lot more than I thought we were going to see. Maybe we should try the experiment. We might see something a little weak. And so Jeanette did the experiment. Here we're comparing the strain that she had made, in which tetanus toxin fragment C is genetically encoded, versus one in which it's been clicked onto the cell surface using the spy catcher spy tag system. And here, not only did we not lose efficacy, we seem to have gained efficacy. Now, for reasons we don't yet understand, we're up at titers of around 10 to the 6, which is very strong in mice. In fact, we're within range of a nanoparticle version of the same vaccine, heavily adjuvanted and injected IM. So it's needless to say, we were all quite shocked to see a response this strong to something that's sort of swabbed on the surface of the skin. Also, gratifyingly, whereas a vaccine administered IM, as you know, is not going to elicit IgA at any of the mucosal barrier surfaces, we find that that this topical vaccine, for reasons I think we're beginning to understand, elicits a pretty strong IgA response, both in the bowel fluid and in the nares. And in fact, I'm not showing this here, a raging IgA response in the intestine from colonization of hair. Okay, so let me wrap up with a couple of slides. First is that just to make a long story short, I think that going back to that original question I started with, I won't overgeneralize, but at least in the case of colonization with staph epidermidis, we had been wrong about the purpose. It is definitely not a homeostatic response. It's nothing weak. And it's not just a local thing. In almost every way, this resembles the immune response to a pathogen with one important difference, and that is that it is preemptive. We believe, and this is a speculation, but we believe the host is reaching out across the skin barrier, grabbing antigen while you're well, bringing it back inside, and developing an antibody response against it, in some sense to prepay the cost of doing so. Again, all the time, preemptively. If that's true, then it's really interesting from a basic science standpoint. It's definitely not what we thought the immune response against colonists looked like. And also, as you could imagine, could be useful. On that topic, we're very interested in whether we could make vaccines this way. Needless to say, no needle, so no need for a healthcare worker. This is something that you could apply to yourself. It could arrive in the mail like in a ketchup packet. For reasons we don't understand, there doesn't seem to be any sign of reactogenicity. Obviously, if that were to hold in humans, then that would also be really helpful. We're getting a robust IgA response at mucosal barrier surfaces, the importance of which I don't need to explain to this audience. And the skin is a big organ, so you could imagine that multiplexing could be done where different skin sites drain to different lymph nodes, so you wouldn't have competition between the two different antigens at the same lymph node. And then finally, I think the component of this that's most interesting right now to me and the group is mechanism. If it's true, as we believe it is, that there is a cell, or there are some cells that are reaching across the barrier, grabbing antigen, bringing it back inside, and developing an antibody response against it, we need to know exactly what is being grabbed and how this works mechanistically. We think we know more about this now, but I think the exciting part about this, in addition to just understanding how it works, is that this would enable us to sneak other things through this channel, maybe even without a bacterial cell at all. And that possibility has us very excited. I'll stop here, thank the folks who did this work, and thank you for listening.
Questions for Michael? Start here.
So this is more of a comment than a question. I really enjoyed your lecture. So I think about 10 or 15 years ago, there was a commentary in Nature or Science by an evolutionary biologist that T-cell memory or immunological memory in general is an adaptation that allowed organisms to harbor commensals. So it's very similar to what you're saying, I think, that it's preemptive. So in other words, for us to be able to have all these commensals in us and they're beneficial, we have to have immunological memory, because if they breach the barrier, we're dead immediately. So I think it fits with what you're saying, if I understood your lecture well. And I'll look it up and send it to you.
Please do. Yeah, I haven't seen it. I mean, my response to that is that, A, that's really interesting. B, it's not the way that we currently talk about in the field. So I think that we felt that, why would you develop an antibody response against staph epidermidis? It's harmless. Like, the denominator is the whole human population on every square centimeter of their body, and it almost never causes trouble if you don't have an indwelling device and you're not immune compromised. But I wonder whether maybe the immune system would say, hey, hold on, this bug would be more than happy to kill you. The reason you're not worried about it is because I've done such a great job preemptively of getting you ready for the inevitable barrier breach when you become bacteremic several times a week. So I think what you're saying is on point.
Great talk. My question is, have you looked at the function of these antibodies? I mean, they're binding. but do they have any effective functions? And I'm wondering whether what these antibodies are doing is maintaining a balance, so that if you were to immunize mu-MT mice, which do not have B cells, with your Staph aureus, and in which case there will not be antibodies, would you find uncontrolled replication of this bacteria that basically takes over the skin?
Yeah, that's a great question. We should do that experiment. You're asking particularly about a function. So I think I had been implying, even though we haven't proven this, that prior colonization protects against of infection and dissemination. And you're suggesting that maybe the role is to limit colonization in the hair follicle, for example, where you see like, yeah, great question. We should do the experiment. I think that there are all sorts of weird phenomena in humans. Neisseria comes to mind, where colonization can be self-limiting, and then all of a sudden it drops over time as people develop an antibody response. So I think that there are examples where we could see this happening, but yeah, I wonder what's going to be the case with staph epidermidis.
Along the same lines, well, what happens with antibiotic treatment? Can you alter it, and does it alter the altered antibody response?
No, that's a great question. We haven't tried that. Part of the reason we haven't tried it is because treatment with antibiotics typically won't clear something like staph epidermidis. So the counts will go down, but then they're already so low to begin with in the follicle that it's hard to know what to expect. So it's something we should look at, but have avoided for aesthetic reasons because of the messiness of the experiment. Yeah, no, you're asking a good question. We should definitely look.
Great talk. I was wondering, do you think it's skin specific or also the gut microbiome that initiates these antibody responses?
Yeah, two comments on that. One is that this is a longer discussion, but we think, and not everybody agrees yet, that in the gut the logic is completely backwards. That everything that the immune system sees that's not a pathogen in the gut is eliciting an antigen-specific Treg response. So it's just default tolerance. You have very broad spectrum tolerance by colonizing an animal. We don't think this is just staph epidermidis on the skin. Other skin colonists and other colonists of oro and nasopharyngeal mucosal surfaces, like from the neck up, all yield something similar to this that we've looked at. So it's not just staph epi on the skin.
Any other questions? All right, let's thank Michael again.